Good afternoon, everyone. It's late, but we've got some really exciting data that we're generating on N-acetyl-L-leucine, and I'll just jump right into it. So many of you guys know my lab is interested in using drug repurposing and the power of fruit flies, which you've heard earlier, to try to find new drugs for rare diseases like SYNGAP1. Last year, I presented this screen that we developed for Syngap1 using the fly eye, where we eliminate Syngap1 in the eye, and you get this abnormal eye phenotype, which you've seen earlier again. And we simply ask, can drugs rescue this phenotype that we see? And we can measure this quite easily, and there's quite a big separation from control wild-type animals. And I showed you that, in fact, we found many drugs that can do this to different extents, and drugs that can make it worse as well, which are also quite interesting. And through kind of talking with the foundation, we settled on one particular drug to follow up on, and there's a ton of other drugs there that we plan to kind of continue with, so we need to have another conversation, Mike and Catherine. But we settled on N-acetyl-L-leucine, which is an acetylated form of the amino acid leucine. You can see that the acetyl group is here. And if you just do a simple public Google search, you'll find that there have been a number of studies testing whether N-acetyl-L-leucine can work in neurodevelopmental or neurodegenerative disorders, in some cases very similar to Syngap1. Last year, we also saw that this was approved as a therapy for Niemann-Pick type C, and that's kind of made things a little more complicated. But many of you know it as Tanganil, which was approved more than 50 years ago in France, and it's been used to treat vertigo. And some of you have tried this, and it seems to be having a positive effect. But we're really interested in kind of trying to figure out why Tanganil works and why N-acetyl-L-leucine works, and I'll kind of get into that in a minute. So we think N-acetyl-L-leucine is actually a prodrug of leucine. It means that the body actually just converts it to leucine once you take it. And the reason why we think this is because leucine, like other amino acids, are highly regulated entry into the cell. The cell only lets in what it needs through the LAT1 transporter. You can't force in more leucine into the cell. However, N-acetyl-L-leucine bypasses the LAT1 transporter, and it's nearly passive transport into the cell through the MCT1 transporter. And so if there's N-acetyl-L-leucine outside of the cell, it will get right into the cell. And within minutes, in cell culture at least, it is deacetylated and converted to leucine. So we think that that's probably what is happening with N-acetyl-L-leucine. So like I said, we want to know why it works and how it works, and we have no idea, right? Like many drug screens and drug repurposing screens, we look for a positive effect without knowing anything about its function. But I think the real driving reason behind wanting to understand is because N-acetyl-L-leucine is not the answer. An amino acid is not a great drug. It's really dirty, has lots of effects, and certainly not all the effects it has are important to Syngap1. And so we want to try to identify what are the important effects, and can we target those better, stronger, and more cleanly. And I think that will result in a better drug long term. So all the work here moving forward is from a really talented graduate student, Haley Tokars, who's here. If you go back in time, you'll see her poster at lunch. But the beauty of flies is that it allows one graduate student to seemingly do 3 graduate students worth of work. So we've taken 2 different approaches to try to figure out what is going on with N-acetyl-L-leucine and Syngap1. A gene expression approach and then a genetic variation approach. And I'll kind of go through both of these. In the first case, we're simply asking how does N-acetyl-L-leucine affect the gene expression profiles in Syngap1 flies. And this is pretty straightforward analysis. We take the heads of these Syngap1 flies that we've generated and do RNA-seq transcriptomic profiling on 4 different conditions: control wild-type animals, drug and no drug, and Syngap animals, drug and no drug. There's lots of different kind of combinatorial analysis we can do here, but I'm just going to present to you the drug-no drug expression profile in the controls, and the same in the Syngap animals. And listed here are the number of genes that are up and down regulated in the control animals and in the Syngap animals by drug effect. And there's kind of 2 really broad observations to make here. First, N-acetyl-L-leucine reduces expression. It doesn't really turn on genes at all. And that was a little bit surprising to us, but you can see that quite clearly here. There are many more downregulated genes. And N-acetyl-L-leucine has a stronger effect in SYNGAP1 animals than it does in wild-type animals. I think it's a moderate stronger effect. It's not that much stronger, but it does kind of increase the number of genes that are differentially regulated in SYNGAP versus control animals. So when we look at the downregulated genes, either in the control animals or the SYNGAP1 animals, the most obvious thing we see is that the proteasome and ubiquitin pathways are completely downregulated. This is a Go analysis on the left, and you can see that there are nearly 60 genes in the ubiquitin proteasome pathways that are downregulated. This is a volcano plot for you connoisseurs showing that all the proteasome genes are down on the left and significantly downregulated. And that's really striking because it doesn't seem to be a SYNGAP-specific effect that we see it in whichever genotype we do this in. And this is a really simplified diagram of the ubiquitin proteasome pathway. Nearly every component you see on this diagram is downregulated by N-acetyl-L-leucine, which is really striking. And for the lay audience, this is basically a pathway that marks proteins that need to be recycled in the cell. They get marked by a molecule called ubiquitin, and then it gets recycled by the proteasome into amino acids back into the cell. So this is really exciting because there's actually a lot of ways to manipulate the proteasome pathway. There are drugs that can manipulate the proteasome pathway. There are approved and lab-grade drugs that can inhibit the proteasome pathway. So we are now throwing all that at the models to see if that's sufficient to get the rescue that we see. There are also lots of genetic ways to inhibit the proteasome. For example, we study another disorder called NGLI1 deficiency, which actually regulates the expression of the proteasome. So now we're looking at genetic interactions between those types of genes and SYNGAP to try to kind of get at this. And again, the fly allows us to kind of throw all of this at the system at once and really kind of get at this. So stay tuned for that. But that was a really obvious common effect in both genotypes. But we also want to know what's specific to SYNGAP that this drug is doing. And again, we see this kind of larger effect, and we see that there are 70 genes that are uniquely upregulated in SYNGAP that aren't in wild-type animals by N-acetyl-L-leucine. Nearly all of these 70 genes are related to stress and immune response, suggesting that the application of N-acetyl-L-leucine on an animal that's missing SYNGAP function results in some kind of stress response. Whether that's positive or not, we don't know, but it could be positive. We also, if we look at the downregulated genes, kind of the 3 times more in SYNGAP versus controls, we see categories like tyrosine and amino acid metabolism, in particular tyrosine metabolism, which the whole pathway seems to be down. And again, this is an amino acid that we're giving to both genotypes, but we only see this metabolism change in amino acids in the SYNGAP animals, not the control animals. There's something about how they deal with amino acids that is specific to losing SYNGAP. We also see genes that are downregulated in energy metabolism, and in particular sorbitol metabolism. Nearly every enzyme in the sorbitol arm of the glucose energy pathway is downregulated, again in a SYNGAP-specific way, suggesting that there is an energy difference in how they metabolize sorbitol, glucose, whatever, in SYNGAP animals versus control animals. And again, there are drugs and genetics that we can throw at all of these, and we are doing all of that Haley's doing all of this simultaneously. Okay, so this is, again, all in progress, but I wanted to kind of give you the other side of how we're trying to get at this, and that's using genetic variation. And this will feel a little bit more different than kind of standard RNA-seq experiments. So we're simply asking, which genetic variants impact the effect of N-acetyl-L-leucine in SYNGAP1 flies, and this goes back to the suppressor question we had earlier in the day, and you'll see that this is actually quite a different approach. So this is really a pharmacogenomics approach at the heart of what we're doing with this particular project, and we're simply asking, and I think that parents here know this, that some patients require high doses of a particular drug, some patients require low doses, some patients require a completely different drug. Some of this obviously has to do with the SYNGAP mutation you have, but also has a lot to do with all the background genetics that's floating around in your genome that's otherwise not affecting your health. And so we want to get at this, and it's really difficult to do in the human population, in particular in smaller groups like SYNGAP1. But we can do this with a fly in the lab quite easily. So we can take our fly model of SYNGAP1 and cross it into a couple hundred different genetic backgrounds in the fly, essentially generating a couple hundred patients with SYNGAP1 with different genetic backgrounds, just like the ones that are here at this meeting. And then we can do the experiment all over again, where we either treat them as controls with no drug or give them drug, just like a clinical trial. And then we can use what we call genome-wide association analysis to identify modifier genes that are driving differences under control conditions, so just basic differences between patients, and also differences between how patients respond to N-acetyl-L-leucine. So this is about 2/3 of the data. We'll have about 1/3 more data when we're done. But this is based on eye size, and you can just kind of see on the left and the right the difference between the worst eye and the best eye. And this data is all ordered by the effect in the control undrugged animals. So there's nearly a 2-fold difference in how severe the eye size phenotype is just by genetic background. They all have the SYNGAP1 mutation, but they just differ by what's floating around in the background in an otherwise healthy animal. And then in blue, you see the effect of N-acetyl-L-leucine on each of those genetic backgrounds. In some cases, they get better on top of the red line. In some cases, they get quite a bit worse, as you can see there on the right side, left side. And in some cases, they don't change at all. And so this is exactly what we wanted to see. This is what we see in the human population. Again, it feels very similar to what you experience as you talk to other parents and other families, right? And so this is exciting, and we have about 1/3 more data to collect. But we can take this data, these differences in the red line and then in the blue line, and ask what variants drive these differences. And this is very preliminary, and we're going to rerun this analysis when it's fully done, so it'll change a little bit. And when we look at the things that are in common between the two conditions, we see the overlapping genes are enriched for things like neuronal guidance, axonogenesis, things that are intimately involved in what SYNGAP1 is doing, which makes perfect sense because that tells us that genetic variation in things that build the nervous system affect SYNGAP1. It makes a lot of sense. But what's really exciting is then we can ask, what are the genes that are driving differences in this red line only, and then driving differences in the blue line only, and I think that will really tell us, what are the pathways that are quite variable, which pathways could we target better using this kind of data, and as we finish the screen, we'll have a lot of that data. And all these genes are suppressors, and all these genes are enhancers depending on which SNP you get in the population. And so this is a ton of modifier gene data that I think will give us years and years of work to do. And I want to end with this huge data dump I'm giving you with kind of one other really interesting analysis that we can do. Part of working with the fly is that the fly community is really good at generating resources and sharing them. This particular resource that we use to cross into the SYNGAP model is used by many labs, and lots of data has been generated on it. And one of the pieces of data that's been generated is expression across all of these genetic backgrounds, just kind of basal, normal expression. And we can take that expression and correlate it with our phenotypes. So I'm going to show you a couple examples. Here, this is a chloride channel in the fly, and it plays a role in neuronal excitability, and it's positively correlated with the eye size in the no drug treatment. So the higher expression this gene is, the better the eye is. And in the drug side of things, this is a negative expression. This is a negative correlation where the higher the expression of this gene called ADG13 is, the worse the eye is. And ADG13 plays a role in autophagy, which has been hypothesized to be not only involved in SYNGAP1, but also part of the leucine function as well. So that's really exciting. So we can kind of pull in this data as well. So I've given you a ton of data, but as you can see, we're using the fly as this kind of unique tool to be able to pull in all this data that we can generate and really tell us, try to uncover every corner, right? And try to get at what the mechanism of N-acetyl-L-leucine by pulling in all this data. And I wanted to finally end with this pretty new piece of data, We can also, eliminate SYNGAP1 in the nervous system using same techniques that we do in the eye, and we can ask whether it affects the nervous function, right? And so when you do this in the fly, the SYNGAP1 animals can't climb as well. So here in black are wild type animals. They get to the top of a vial in less than 30 seconds. It's pretty standard, pretty good. But SYNGAP1 animals, fewer than 60% ever make it to the top, and they kind of plateau, bottom out. We've talked about plateau a lot, right? And so we asked, can we raise these animals that don't have SYNGAP1 function in the nervous system, and what does it do for their climbing? And it improves their climbing quite a bit, actually. And this effect in climbing is stronger than the effect that we see in the eye by N-acetyl-L-leucine. So this is exciting. We've only done a single dose, and so we can push this dose quite a bit higher. And I think it'll be really exciting to take the observations that we're trying to find, the mechanistic observations we're making with all the other data, and throw it at this model, which I think tells us that N-acetyl-L-leucine can also work in the central nervous system as well. So again, I just want to thank Haley's done all the work on this project, and Cure Syngap for providing funding for this project, and I think it's money that's been well invested. Thank you.